Assalamu alaikum dear students. Today we will discuss regarding how to label the nucleic acids. The labels could be isotopic or non-isotopic, which means they could either be radioactive or non-radioactive. When we talk about non-radioactive traces, they could either be fluorescent or the chemiluminescence ones. Fluorescence means colored and chemiluminescence means with the light, जिसको आप लग से मैयर करते हैं. And we can label either our nucleic acid at the terminus, 5' terminus or the 3' terminus, or we can put these labels internally within the chain. And as anticipated, because nucleic acids are of two types, DNA and RNA, the probes or the labeled nucleic acid which we will synthesize will either be DNA or the RNA based ones. And when we talk about probes, probes are the single stranded 18 to 22 nucleotide long DNA sequences which are used for hybridization reactions, and we utilize them for identifying our genes of interest or the sequence of interest. So when it comes to the choice of the labels, whether to choose a radioactive label or non-radioactive label, it is important to see what benefits they possess and what are the disadvantages. So when it comes to radioactive labels, they are highly sensitive, but at the same time, they are very dangerous for health because of their mutagenic properties, and they are also short-lived. So you have to resynthesize them again. But when it comes to the radioactive labels, their benefits include they are safer, they are not dangerous for your health, have longer shelf life, but at the same time, the sensitivity is very low as compared to the radioactive labels. So, thanks to the radioactive tracers, there had been tremendous discoveries in the field of biochemistry and molecular biology. So, they had been used for quite a long time. And the procedures for radioactive, tracers were well established. But now they are not in use, as you all can, figure out very easily कि चूंकि वो health hazards हैं, so they are no longer used. Anyhow, in the past, when they were used, the most common isotopes were tritium, carbon-14, sulfur-35, and phosphorus-32. We can classify them as low energy emitters, medium energy emitters, and high energy emitters. Among the low energy emitters are the tritium and the carbon-14, whereas the medium energy emitter are sulfur-35 and the high energy emitters are phosphorus-32. Our tritium, carbon-14, and sulfur-35 were used for direct autoradiography, and P32 was used for indirect autoradiography because of its high energy emitter property. This P32 is more hazardous than any other isotope and requires particular care in use, and there were strict statutory requirements for the storage and disposal of such radioactive waste materials. Partly because of the inherent dangers of working with high energy isotopes, uh, the scientists looked for alternative technologies. And the alternative technologies for labeling include fluorescent dyes or enzyme linked labels, which we will discuss shortly. When it comes to radioactive labels, they decay with time, and ultimately, it is necessary to synthesize fresh probe before each experiment because of their short shelf life. Although radioactive labels are not used anymore, but being a biochemistry student or a biotechnology student, we should know about this obsolete technique. Labeled nucleotides may be counted in a scintillation counter to determine the amount of nucleic acid present. So this is the specified instrument, what we call it as scintillation counter. When we do the calculation, we take into account the amount of radioactivity present in the sample, because that will directly relate to the amount of nucleic acids. The difference between labeling for the tracing purpose, and labeling for the probes is largely dependent upon the specific activity. And what is specific activity? It is simply the measure of how radioactive the molecule is. So when it comes to tracing, we used to check we used to have low specific activity, but for probes with high specific but for hybridization reactions, high specific activity probes were necessary. In probe preparation, the radioactive label was usually the P32, which was a high energy beta emitter. Why? Because it can travel farther. As a result, it can spread the signal. Whereas when it comes to sulfur 35, it gives morphological resolution, and that helped in studying expression of the genes and cells in the tissue. So there are different uses for different type of radioactive probes. This table tells us the characteristics of radioisotopes for labeling nucleic acid. You can see the tritium, phosphorus 32, phosphorus 33, sulfur 35. You can see their half-life, and among them, the shortest half-life is of phosphorus 32, which is 14.3 days, whereas the longest one is of tritium, which is 12.4 years. Their decay type is beta, and their emission energy values are also given, whereas exposure time has been shown to, along with the suitability for high resolution studies. And you can see tritium is excellent for high resolution study, whereas phosphorus 32 is very poor, and we have discussed in the last slide the reason for its poor nature and its poor suitability for high resolution studies because of the dispersal of the signal. So autoradiography is something similar to the X-ray monitoring or X-ray documentation. What happens? It records the position of a radioactively labeled compound within a solid sample by producing an image in a photographic emulsion. What we do, we place the solid sample with an X-ray film. X-ray film, as we have seen already, we all are very well aware of that it is a plastic sheet with coating of photographic emulsion, and the photographic emulsion is silver halide suspension. So what happens, that radioactive emission travels through emulsion, and during that traveling it converts the silver ions into silver atoms, and as a result it gives the two-dimensional representation. When we add the radioactive atom into the nucleotides, it can either be done via external labeling or internal labeling. External labeling is when we add to one end of the probe, and internal labeling is when we insert the nucleic acid within the chain. You can see here the position for radioactive P32. It can either be on the alpha phosphorus or the beta phosphorus or the gamma phosphorus. When we want this P32 to get incorporated within the growing chain, so it is very important to place our radioactive P32 at alpha position. So for internal labeling, the alpha phosphorus should be radio labeled. But when we are doing the external labeling, then it should be the gamma phosphate, uh, which should be the radioactive one or the P32 one, because then it will be present at the terminal and it won't be removed while in getting incorporated into the chain. As we all are aware of that both beta and gamma phosphate, they remove from the nucleotide in the form of pyrophosphate. So only alpha phosphate gets incorporated into the growing chain. Now we are going to see certain techniques which are utilized for the labeling purposes in the genetic engineering or molecular biology labs. The first one is end labeling. For end labeling, we use a special enzyme known as polynucleotide kinase. I have discussed with you earlier that kinase is that enzyme which is responsible for the shifting or the transfer of phosphate molecule from a donor phosphate molecule to an acceptor molecule. So here you can see at first what will happen. We will have a donor phosphate molecule with the terminal p32 phosphate, and that terminal p32 phosphate will be replaced with the help of polynucleotide kinase on the probe. यहाँ पर आप बहुत क्लियरली देख सकते हो कि जो ऑलरेडी एग्जिस्टिंग प्रोब पर फॉस्फेट मौजूद था वो ब्लू कलर का था, वेयर एज द पीच वन वॉज द रेडियो लेबल वन, और वो एटीपी के टर्मिनल पे मौजूद है. सो दिस polynucleotide kinase has actually replaced the blue phosphate with the peach one, and the peach one is actually the radio labeled one. On this particular slide, you can see the internal labeled positions, and for both cases, you can see it is the alpha phosphate. But when it comes to the second position, the second nucleotide, there you can see instead of phosphorus, you can also replace one of the oxygen at the alpha position with sulfur 35. So we can utilize these two ways for internal labeling. In this slide, you can see how polynucleotide kinase can be used for replacing, for actually inserting a phosphate into a DNA molecule, and this phosphate is coming from a donor molecule which is ATP, and again the terminal phosphate is the labeled one. The second technique for labeling purposes is nick translation. In this particular technique, we first create a nick. Nick is a missing phosphoester bond. I have discussed with you earlier in the previous lectures the difference between nick and a gap. So nick is a missing phosphodiester bond, whereas gap is missing nucleotide or nucleotides. So first we generate the nick with the help of DNase 1, and then we will utilize DNA polymerase 1 to actually uh, put nucleotides in. What happens when this polymerase 1 it starts adding nucleotide, तो ये क्या करेगा, वन बाय वन पुराने वाले को निकालता जाएगा और उसके रिप्लेसमेंट में न्यू वाला इंसर्ट करता चला जाएगा. और जब ये न्यू वाले इंसर्ट होंगे तो उनमें हम रेडियो लेबल्ड न्यूक्लियोटाइड्स चूँकि हमारे सोल्यूशन में मौजूद होगा तो वो इनकॉर्पोरेट होता चला जाएगा. एंड व्हाई दिस इज हैपनिंग? बिकॉज ऑफ द 5' to 3' exonuclease activity of the polymerase which we are using. We all are aware of that polymerases possess a very excellent 5' to 3' polymerization activity. But the polymerase can also possess 5' to 3' exonuclease activity and 3' to 5' exonuclease activity. In this particular case, the E. coli DNA polymerase 1 which was used possesses 5' to 3' exonuclease activity. So just like a bulldozer, whichever nucleotide is coming in front of it, it will take it out, and as a result the gap will keep on generating, which will be filled via the polymerization activity of the same enzyme in 5' to 3' direction. And what will happen at the end? One nick will remain, and that nick will be sealed with the help of DNA ligase. The third technique is labeling by primer extension, and I consider it one of the very easy techniques. It is very clear from this slide that at first we will take the DNA, we will increase the temperature and will turn it into single-stranded DNA, and after that we will make use of a primer to hybridize with this single-stranded DNA. Once hybridized, this primer will ultimately be extended via DNA polymerase. and we will provide the radio labeled nucleotide in the solution. So when based upon complementarity, the DNA polymerase add the fresh nucleotide to the growing chain. It will incorporate the radio labeled one. So in this way we can do the internal labeling. अगेन एन एक्सेलेंट स्लाइड बहुत अच्छा कॉन्सेप्ट इसमें नज़र आ रहा है द रैंडम प्राइम्ड डीएनए लेबलिंग you can see इसमें double stranded DNA है first we have to denature it we all are aware of for denaturation we use temperatures 94 to 96 degrees centigrade they will turn into single stranded DNA then we will use these random primers now you will must be interested in knowing what are random primers. Random primers are the short sequence primers. They are around 5 to 7 nucleotide long. Because they are very short, so they can bind at random sites close to each other. अब इसके लिए आप एक फॉर्मूला याद करें जो मैंने आपको करवाया था 1/4 n तो जब प्राइमर का साइज अगर 5 न्यूक्लियोटाइड लॉन्ग है तो हमारा फॉर्मूला हो जाएगा 1/4 5 which means 4 multiplied by 4 multiplied by 4 multiplied by 4 multiplied by 4 so आपके पास जो नंबर आएगा that will be a small number which will be close to each other एज अ रिजल्ट आप जब इस रैंडम प्राइमर को हाइब्रिडाइज कराएंगे तो ये अपने इस सिंगल स्ट्रैंडेड सीक्वेंसेस पर रैंडम जगहों पे और काफ़ी ज़्यादा जगहों पर अटैच हो जाएगा after that we will make use of the clenos subunit of DNA polymerase and the solution of the DNTPs with the labeled nucleotides present within them for the primer extension and when it will extend it will cause the internal labeling of the nucleic acid which is DNA in this particular case. Another way of doing it is end filling. Carefully look at the figure. टॉप पे आप देखिए आपने एक डीएनए फ्रेगमेंट लिया and then you will digest that DNA fragment with a suitable restriction endonuclease. In this particular case, they have used EcoR1 and as a result of cutting, 5' overhangs were produced. And after that generation of overhangs, they will be filled again. The ends will be filled again with the help of the DNA polymerase, clenofragment, and the labeled nucleotide. You can see when the end will be filled based upon complementarity where T's are present, A's will be added and the A's provided in this solution are the labeled ones. Sorry, not the radio labeled, the labeled ones. So when they will fill, you will have the DNA with the terminal, not actually terminal, but because they are present towards the end, so I'm saying terminal. In reality, we call terminal as to those nucleotides which are present at the extreme rear end. But nevertheless, they are also present towards the terminal. So we will get the labeled end labeled DNA. We can then again cut it with restriction endonucleases with the internal cleavage site to generate different sized fragments, the different sized labeled fragments. Because we were discussing DNA labeling, it is also very important to discuss RNA labeling as well. Because at times we are interested in labeling the messenger RNA products or their complementary sites on DNA. So what we are going to do for this purpose हम लोग एक वेक्टर लेंगे यहाँ पे आपको जो वेक्टर नज़र आ रहा है this is PSP64 and this is an expression vector as is evident from the presence of the promoter site. You can also see the origin of replication, the second important thing as multiple cloning site, and the third important thing in the form of selectable marker, which is ampicillin resistant marker in this particular case. अच्छा आप देखो इसमें जो sp6 प्रमोटर है उसके बिल्कुल नीचे मल्टीपल क्लोनिंग साइट मौजूद है. So what you will do, you are going to digest it with your required or the suitable restriction endonuclease. After cutting, you will insert your gene of interest against which you want to prepare labeled RNA. जब आपकी वो जीन इंसर्ट हो जाएगी तो अब आप उसकी जो स्पेसिफिक आरएनए पोलीमरेज है इस वेक्टर के लिए which is sp6 RNA polymerase, you will make use of it, and you will also provide the nucleotides for the RNA synthesis, which are ribonucleotides, and among them it is the UTP which is labeled. So by the end of this experiment, you will be having nested fragmented RNA labeled molecules which you can utilize for different purposes. Having said this, now I want you to show the non-isotopic labeling, which could either be done via direct method or indirect method. So let me show you the figure first. Here you can see the fluorescent labels in the form of fluorescein, which gives green color, rhodamine, which gives red color, and aminomethylcoumarin, which gives indigo color. So you can attach these fluorescent dyes to your nucleotide via spacer molecule, as is clearly evident on this slide, which has been highlighted with the gray or the dull blue color. अच्छा अब जब आपने ये फ्लोरेसेंट लेबलिंग कर ली है तो हाउ यू आर गोइंग टू विजुअलाइज इट फॉर दैट यू विल बी इन नीड ऑफ फ्लोरेसेंट microscope. So fluorescent microscope will have excitation filter, diachronic mirror, and then it will direct the light towards the objective, and after that it will reflect back via diachronic mirror towards the reader through बैरियर फिल्टर. इस तरह आप कलर्ड सिग्नल्स को आराम से चेक कर सकते हैं. यहाँ पे देखें आपको एक बड़ा इंटरेस्टिंग एस्पेक्ट दिखाया जा रहा है of the labeling of DNA. You can see here कि आपके पास एक डबल स्ट्रैंडेड DNA मॉलिक्यूल अगर मौजूद है तो you can first label it with the reporter group, and you can do it via inserting or via providing the cell with the labeled nucleotide for their in vivo replication purposes. Anyhow, once done, you can see one of the strand contains nucleotides with the reporter dye or signal to it. Now this reporter dye has higher affinity for uh, the molecule, an affinity molecule, and that affinity molecule, which is shown here in green, is in turn attached to the marker molecule. So wherever the reporter nucleotide is, the affinity molecule will go and bind to it, and with affinity molecule the marker is attached, so that will give fluorescence, and we can detect it via the fluorescent microscopy. You can see here that the DNA labeled probes can be labeled with the help of biotin UTP, and you can see that in the growing chain, one of the chains has been replaced with the modified nucleotides attached with biotin. So afterwards you can utilize some probe which can specifically bind to this, or you can utilize this as a probe to identify the gene of your interest. You can do it for the sake of diagnostic purposes, or you can do it for the sake of research purposes, both. So यहाँ पर आप देखें आपके पास जो सैंपल पेशेंट से एक्वायर किया गया है उस पेशेंट उस सॉरी जो सैंपल आपने पेशेंट से एक्वायर किया था उसको आपने फिक्स किया हुआ है ऑन अ सॉलिड सरफेस, और अब अपनी जीन ऑफ इंटरेस्ट को आइडेंटिफाई करने के लिए आपने प्रोब्स, लेबल्ड प्रोब्स को सिंथेसाइज किया है। एनी हाउ, ये जो बायोटिन लेबल है इसको आप डायरेक्टली नहीं देख सकते, सो यू विल एड अ मोलिक्यूल which is evident, which will go and bind to the biotin. ये शायद मैंने आपको पहले ही बताया है, आज मैं रिपीट कर रही हूँ कि द मोस्ट फेमस कपल इन बायोकेमिस्ट्री इज बायोटिन एंड स्ट्रेप्टोबाइडिन। स्ट्रेप्टोबाइडिन का को सिंप्लीफाई करके आप एविडिन भी कह सकते हैं। ठीक है, स्ट्रेप इंडिकेट करता है बैक्टीरिया को, स्ट्रेप्टोकोकस को। सो वो एविडिन प्रोटीन जो कि स्ट्रेप्टोकोकस से आइडेंटिफाई की हो, आइसोलेट की हो, उसको आप स्ट्रेप्टोबाइडिन कहते हैं। ये एग में भी मौजूद होती है। एनी हाउ, सो जहाँ भी बायोटिन होगा वहाँ पे एविडिन आकर बाइंड करेगा, और उस एविडिन के ऊपर आप फ्लोरेसेंट मार्कर्स को अटैच कर सकते हैं ताकि आप उसके सिग्नल को रिसीव कर सकें वाया फ्लोरेसेंट माइक्रोस्कोपी। Having said this, look at this labeling with Horsch reddish peroxidase. You have the single-stranded DNA polymerase. आप इसमें पहले insert करा देंगे Horsch reddish peroxidase along with the glutaraldehyde, and after that you will let it hybridize uh, with the with the fixed DNA. अब इस पर्टिकुलर केस में द फिक्स्ड डीएनए कुड बी ऑफ द डायग्नोस्टिक वैल्यू और इट कुड बी फॉर द रिसर्च पर्पसेस। एनी हाउ, ये जो हमारे पास लेबल्ड प्रोब था इसमें हॉर्स रेडिश पेरोक्सीडेज मौजूद थे जो कि बाइंड कर सकता है ल्यूमिनॉल के साथ, और वो ल्यूमिनॉल अल्टीमेटली लक्स प्रोड्यूस करेगा या लाइट प्रोड्यूस करेगा। सो वी कैन कैप्चर द chemiluminescence light signals for it. Again, here you can see the nucleotide with the spacer molecule and a reporter dye, or the इसको आप dig oxygen reporter group भी कह सकते हैं, and the spacer is 11 carbon long. The second one is the biotin 16 UTP1, where the reporter group is biotin and it has affinity for, as I have told you, streptavidin या फिर avidin. Now, towards the final slides, it is important to determine what labeling system will work best in a given application, and that is dependent on the size and type of the nucleic acid to be labeled and the choice of the label required for the application. So with this, we are at the last slide of our talk, and this is comparison of the labels. We have discussed each and every point related to our labels, and you can see it has been summarized here. Keep also in mind that the cost for radioactive labels is high because it requires frequent renewal, whereas for non-radioactive it is lower. With this, we are done for today's lecture. Best of luck. Must go through it, and if you find it difficult, you can discuss it with me.